Yes. So in the last class, we were discussing kingdom plantae, and yes, in the kingdom plantae, we have finished with halophytes. We have done. Yes. So today we will discuss. Yes, four divisions we have to study: bryophytes, pteridophytes, gymnosperms, and angiosperms. Yes. So today we will discuss angiosperms. If you understand angiosperms, bryophytes, pteridophytes, and gymnosperm will be easy to understand. That clear? Yes. Why? What is the reason, sir? Because in the angiosperms, when we talk about the angiosperms, first thing we need to remember is here the angiosperms are flowering plants. Okay, flowering plant. So uh, first we will go with the parts of the angiosperms flower. Then we'll go with the characteristic features. Yes. So when we talk about the angiosperms, they are called as flowering plants because flower is the reproductive part of an angiosperm. So here, when we talk about the flower consists of how many parts? Any idea? How many parts are there in a flower? Can you recall? You have studied in first year PUC. Huh? Consists of how many parts? How many whorls? Whorls, huh? Are parts of a flower. How will you differentiate? How will you separate? Yes, one part, two, three, four parts. Yes, sure. Can you name them? Can you name them? Yes. You have not seen a flower? Yes. Calyx, corolla, yes. Androecium and gynoecium. Four whorls of the flower. Calyx, also called as sepals, and corolla, also called as petals. Yes. Androecium, male reproductive part, and gynoecium, female reproductive part. So here, when we talk about the calyx and corolla, they are called as accessory parts, and androecium and gynoecium are reproductive parts. Did you understand? Yes. What is a flower? We know. Flower is the reproductive part of an angiosperm. So all the flowering plants which you see that belong to the division angiosperms. So when we talk about the flower, how many valves are there in a flower? Totally four: calyx, corolla, androecium, and gynoecium. Calyx also called as corolla, also called as petals. Androecium, gynoecium, female reproductive part. Calyx and corolla also called as accessory part. And androecium and gynoecium, what do you mean by accessory parts? Yes, reproductive part we will understand. Yes, the part which is involved in the reproduction is called as reproductive part. What do you mean by accessory parts? Yes, supportive. Supportive parts which are supporting the reproduction process. Is that clear? Yes. Any doubts you have till here? No doubts. Everything is clear. Yes. So here, when we talk about the angiosperm, we need to, yes, we need to study about the two very important part, that is the androecium and gynoecium. Yes. So as I told you, androecium is the male reproductive part. So here, this androecium is composed of many stamens. It is composed of many stamens. If you take the structure of the androecium part, that is one stamen. If you take the single stamen, so here this one whole structure is called as stamen. Complete. So like this, if it is 10, 20, 100, all together makes androecium. Okay. Now again, this stamen consists of two parts. How many parts? Two parts. Okay. This is one part and this is one part. Okay. So the long slender like structure, no, which is giving the standing platform, it is called as filament. It is called filament. And the reproductive part, where the reproduction process takes place, production of male gametes takes place, it is called as anther. Yes, it consists of how many parts? Two parts. One is anther and one more is filament. Did you understand? Yes. Male androecium is the male reproductive. It is the male reproductive part. So it consists of many stamen, and each stamen consists of two parts. One is anther, one is filament. Did you understand till here? Any doubts? Shall we proceed? Yes. Now, so here, when we are studying about the angiosperms and when we are studying about flower, the major thing we need to study is about the anther. Yes. So here, if you cut open this anther, if you are cutting the anther, we will be able to see the transverse section of the anther, like this. Transverse section of the anther. TS of this structure is called TS of anther. It is called TS of anther. So here, when we study what is present inside, which is responsible for the production of male gamete, here we are able to see four structures. Okay. One anther, if you take one anther, it has two lobes. This is one lobe This is one lobe, this is one lobe. So anther is bilobe. ठीक है? Anther is bilobe. ठीक है? And one lobe, if you take this as one lobe, ne? In this, this structure is called one theca. It is called. So anther is bilobed with two theca. Anther is bilobe with one lobe, two theca. Two lobes. ठीक है? Did you understand? Ditheseous we will tell. Ditheseous. That means one lobe is having two theca. Is it clear? Now, so here, if you cut the transverse section of the anther, we are able to see four structures inside. Okay? That four structures are called as microsporangia. Yes, this if you are studying second PUC now, I think you have already studied in the first chapter. Yes. So microsporangia. Is that clear? Yes. Now, from outside, it is covered by how many layers? How many layers are there? 2, 3, yes, totally 4 layers. The anther is covered by how many? 4 layers. Yes. The outermost layer is called epidermis. Then next layer is called endothelium. Then next layer, middle layer. Done? Yes. Now innermost layer is called tapetum. So how many layers we are able to see? 4 layers. When we talk about the function of these 3 layers, epidermis, endothelium and middle layer, they are mainly meant for protection. They are outermost covering. Is it clear? Now coming to the tapetum, what is the function of tapetum here? Yes, it will nourish. So nourishment. Done? Did you understand till here? Yes. Now coming to the microsporangia. Inside the tapetum we have microsporangia. So here inside the microsporangia, when the anther is young, no, it will have small tissues. It will have small tissues. The tissues are called all are second PUC here. Yes, second PUC students. Pollen grains, very good. Directly pollen grains will come. Huh? Yes. Connective tissue. Connective tissue is present in between. In the center it is present. Okay. I am asking you inside. Connective tissues are present in between, because of which all the 4 microsporangia are holded together. Okay. What is present inside when the anther is young? Directly pollen grains will come. No. Then what is present? Don't know. Sporogenous tissues. What are present? Sporogenous tissues are present. Now, this sporogenous tissue, no, when it undergoes meiosis, when it will undergo meiosis, so that time it will become microspore mother cell. Did you understand till here? Yes, we started from Androecium. Androecium composed of stamen. Stamen consists of two-part anther and filament. When you open the anther, no, inside the anther we have microsporangia. Inside the microsporangia we have sporogenous tissue. This sporogenous tissue undergoes the meiosis. Okay? It is a diploid cell. So it will undergo meiosis to form microspore mother cell. Done? Did you understand till here? Now, each microspore mother cell, huh? Next, it will undergo a process called microsporogenesis. Yes, each microspore mother cell. If you take one microspore mother cell, each microspore mother cell will undergo meiosis again, no, by a process called microsporogenesis to form how many? To form, yes, each microspore mother cell will produce how many? Four microspores. So when it is four microspores, it is called microspore tetrad. Did you understand till here? Yes. Now, this microspores, when the anther is matured, no? When the anther is matured, so dehiscence of anther takes place. Means opening of the anther, dehiscence of anther, layer by layer, the anther will open. All the three productive layer will open. Then the tapetum will open. Then the microsporangia will open. So microspores will come out. When the microspores are present inside, they are called as microspores. The same microspores, when they come out, no, when they start shedding, they are called as what? They are called as pollen grains. Okay? Did you understand? Yes? Any doubts you have till here? Everything is clear? Yes. Now, what do you call a pollen grain? What do you call? What is a pollen grain? If I ask you, what you will tell? Hmm? Yes? What is a pollen grain? All are from second PUC only now. Yes? First chapter is over. Which chapter is going on? Huh? Second chapter, third, fourth, fifth, fourth chapter is going on. Huh? Yes. First chapter, don't know. Right? We have listened from here, we have passed on from here. Finish. Chapter close. Yes? That is the case. सुने भूल गए कहानी खत्म रात गई बात गई नहीं yes or no yes any doubts still here any doubts still here no doubts yes androecium is composed of stamen consists of anther and filament yes so anther how many lobes are there how many theca four theca ठीक है one lobe two theca therefore dithecious in nature done did you understand yes now how many layers are there to the anther four layers outermost layer is called second layer third layer innermost layer what is the three layers doing outermost layers protection ठीक है innermost layer nourishing the porogenous tissues then nourishing the microspore mother cell then finally nourishing the microspores and pollen grains is that clear did you understand yes inside the microsporangia what is present when the anther is young porogenous tissues are present okay that porogenous tissue when they undergo meiotic division they will convert into microspore mother cell when the microspore mother cell undergoes meiotic division which process takes place what is microsporogenesis yes tell me what is microsporogenesis The word itself is telling you, no. See, Genesis means formation. Genesis, meaning of the word Genesis is formation. Process of formation of microspores. Okay. So one microspore mother cell will give rise to four microspores. That four microspores together they are called as microspore tetrads. Okay. When the dehiscence of anther takes place, the microspores come out. Okay. So when the microspores are shedding, what they are called? They are called pollen grain. Now, coming to the point, what exactly is pollen grain? In one word, if I ask you, what you will tell? What is a pollen grain? Shall I erase? Shall I erase this? Yes? Shall I erase? Yes. If I ask you what is a pollen grain, tell me. It's a gamete. Oh, very good. Any other answer? Yes? Single cell. Very good answer. Huh? Pollen grain is a male gametophyte. It is called a male gametophyte. Now, the question arises, what is a gametophyte? You should know basic things. Nay? Yes? What is a gametophyte? Now, if you study bryophyte, again there you will study the plant body is a gametophytic in nature. If you study gymnosperms or pteridophyte, there the plant body is sporophytic in nature. What is this sporophyte? What is this gametophyte? That you should know. Nay? Yes? Yes? Gametophyte, gametophyte is a cell, okay, where gamete formation takes place. Okay? Inside the gametophyte, gametes will form. Is that clear? Inside this gametophyte, gametes will be formed. Yes. So when we talk about the pollen grain, which is also called as a male gametophyte, okay, how many cells we are able to see? Any idea? Yes. In pollen grain, you have studied the structure of pollen grain. You have studied? Yes. You have done practicals? No. Nobody have done practicals. You have not lively seen the pollen grain. Yes. So tell. When we talk about, listen here. This is one of the very important topic in the second PUC also in the first PUC, because at least two questions will be asked in the CET and NEET related to angiosperms. Okay. Here, in the theory also, in the second PUC first chapter, very important. Practical also, last year, this year, 2025-26 question paper, 2 mark question, draw a label diagram of pollen grain. So, one question will be related to this chapter. Okay. In practical also, almost 1, 2, 3 practicals which you will perform in the final exam is related to this topic only, angiosperm. So careful. So here, when we talk about the pollen grain, first thing we got to know that it is a male gametophyte. Okay? And this male gametophyte, this pollen grain is microscopic in nature. It is microscopic in nature, means we are not able to see through our naked eyes. If you want to see a pollen grain, we should be able to see with the microscope. Is that clear? Why, sir? So here, if you see, take a flower. Okay, take a hibiscus flower or rose flower or vinca flower. Any flower you take, you are able to see yellow color spots. If you take a rose flower, right? So that which we are able to see, it's a pollen sac. It is a pollen sac. Inside the pollen sac, microspores will be present. That microspores, when it comes out, it becomes a pollen grain. Is that clear? So number one, male gametophyte. Number two, microscopic in nature, measuring about 25 to 50 micrometers in diameter. Okay, 25 to 50 micrometers in diameter. Done. Yes. Now, two very important things here. When we talk about pollen grain, we need to study the pollen grain consists of two layers. Consists of two layers, and it consists of two cells. Two very important things. The pollen grain consists of two layers and two very important cells. The two layers are outermost layer, innermost layer. No? So here the outermost layer is exine. Outermost layer is exine and innermost layer is intine. Done? Did you understand? Now coming to the two cells. Two cells we have, one large, one small. Okay? That large cell is called vegetative cell. The small cell which the pollen grain has, it is called reproductive, or another word we can use, generative cell. Done? Here this intine layer is made up of pectin or cellulose, and the outermost layer is made up of a hard chemical, a very strong chemical that is called as sporopollenin. Done? Yes. So here when we talk about pollen grain, point number one, they are male gametophytes. They are microscopic in nature. Yes, measuring about 25 to 50 micrometers in diameter. Yes, it has two layers. It has two cells, two layers. Outermost layer is called as exine. Innermost layer is called as intine. And two very important cell. One is called as vegetative cell. One is called as reproductive or generative cell. Yes, the outermost layer consists of a strong chemical that is called as sporopollenin, and innermost layer is made up of cellulose or pectin. Yes. So here when we talk about the outermost layer, this is like a shield, a protective layer. Because of the sporopollenin, the pollen grain can withstand, it can withstand high temperature, enzymes, acids, strong alkalis, etc. Nothing will happen to the pollen grain. Yes, if you see the structure of the pollen grain, we will be able to see the structure of the pollen grain with two layers. One is called as the exine and intine. Here inside we have a huge cell that is irregular in shape, vegetative, and a small cell that is generative cell. Yes. So here question arises, what is the function of vegetative cell and what is the function of generative cell? Cytoplasm is present both in the vegetative cell as well as the generative cell. The generative cell is spherical or oval in shape. Okay? The vegetative cell is irregular in shape. Is that clear? It is large, irregular in shape. Vacuole is also found with the cytoplasm. Yes. Here the question arises, what is the function, why these two cells are found in the male gametophyte, vegetative cell, generative cell? Can anyone tell me? Yes. The function of the male gametophyte is to produce gametes. Nay. No, which cell will produce gamete? Vegetative cell will produce or the reproductive cell will produce? Yes. It is vegetative. Yes. See, vegetative cell, no, is mainly responsible for storage of food and providing nourishment to the generative cells. Okay? Because it is large, food is stored. I told you, vacuole is present. What is the function of vacuole? Storage part. Right? So, vegetative cell mainly meant for storage of food and providing nutrients, nourishment to the developing cell. And generative cell, yes, it is mainly responsible to produce two male gametes. How many gametes will be produced? One pollen grain will produce two male gametes. Is that clear? Did you understand from where we started? We started from androecium. Androecium composed of stamen, stamen composed of anther, inside the anther microsporangia, inside the microsporangia sporogenous tissue. That sporogenous tissue will get converted to microspore mother cell. Microspore mother cell will produce microspore and microspore will get converted to pollen grain. Inside the pollen grain we are able to see two cells. One is vegetative, one is generative. Vegetative cell is storing food for the nutrition and the generative cell or the reproductive cell is producing two male gametes. Done? Did you understand? Yes. Now, one more thing we need to see here. If you see the outermost layer, right? If you see the outermost layer, there in some points there is a cut. Yes. Yes. What is that called? Any idea? No idea. Hmm? See, pollen grain has produced two male gametes. Nay, whatever, if you take the microscopic structure also, Almighty Allah has made everything with the perfection. Nay, male gametes are produced. If you understand this point, male gametes are produced. Where they are produced? Inside the generative cell. Nay, yes. Now, if you take the outermost structure is made up of sporopollenin. What I have written? A hard chemical, strong chemical. Nay? It can withstand high temperature. If you take that pollen grain, put in the high temperature, nothing will happen. If you put in the acid, nothing will happen. If you put in the chemicals, enzymes, alkalis, nothing will happen. Nay? Then, how this male gametes have to come out? The male gametes have to come out to fuse to undergo the next process that is called fertilization. Yes or no? No, how they have to come out? Yes, yes. So I have marked, so from there it will come out. Since I have marked circle, from there it will come. See, wherever I have marked the circle, no, there sporopollenin is absent. See, from here till here, sporopollenin is present. Okay. Here also it is absent. Here also it is absent. Then, where sporopollenin is absent, no, where sporopollenin is absent, that part of the pollen grain is called germ pore. एक होल बोलती नहीं, छोटा होल, होल के पॉइंट के सर का, तो वैसे पॉइंट्स आती, that is called as germ pore. Did you understand? Yes. When the pollen starts germinating, जब pollen grain ना germinate होना शुरू होता है, grow होना शुरू होता है, ने? So here what happens? Since the innermost layer is made up of cellulose, ne? इन्हें थिन रहता है, ठीक है, पेक्टिन थिन रहता है, काइटिन थिन रहता है, थिन केमिकल कंपोनेंट वो, तो इन्हें ओपन हो जाता है, ठीक है, दिस विल ओपन, ठीक है. नेक्स्ट, here there is no hard chemical, pore like structure is present, so the male gamete which is released will come out. So how it will come out? Directly it will not come out. So here wherever the point spore germ pore is present, a tube like structure will be produced like this. ऐसा ट्यूब बनता, उसे हमें पोलन ट्यूब कहते. तुम्हारा एक्सपेरिमेंट है, ठीक है. एक्सपेरिमेंट नंबर 2, कैलकुलेट द नंबर ऑफ पोलन जर्मिनेशन, परसेंटेज ऑफ पोलन जर्मिनेशन बोल को, ठीक है. सो हियर व्हेन द पोलन स्टार्ट्स ग्रोइंग, ऐसा ट्यूब आ जाता है, ठीक है. सो फ्रॉम दैट ट्यूब व्हाट हैपेंस, द मेल गैमेट विल ट्रैवल एंड कम आउट. Is that clear? Did you understand? Yes. From the androecium till the male gametes, whatever we have studied, it is about the androecium, male reproductive part of the flower. Did you understand? Yes. Shall I erase? Shall we proceed with the female now? Yes. Yes. Gyno means female. Okay? Gyno means female. Gynoecium. So here, when we talk about the gynoecium, this gynoecium is composed of carpel or pistil. It is composed of carpel or pistil. If you take a hibiscus flower and if you cut the center part, if you remove the center part, you will be able to see the structure like this. This complete structure, no, is called as carpel or pistil. We can call it as carpel or pistil. If it is one single, it is called monocarpellary. If it is more than one, bicarpellary, tricarpellary, tetracarpellary. How many are there? We can tell. Okay? If it is more, multicarpellary also can be told. Is that clear? Did you understand? Yes. Now, this carpel or pistil consists of three parts. It consists of three parts: stigma, style, and ovary. Is that clear? Did you understand? Yes. Gynoecium composed of carpel or pistil, and each carpel or pistil consists of three parts: stigma, style, and ovary. Here, when we talk about the stigma, it is called as the landing platform. It is called the landing platform. For what? Yes, for pollen grains. The pollen grains, when it is shed, when it reaches, it will come and sit here. It is the landing platform. The style is just the supportive part, which is supporting both the stigma and ovary. Okay? And the ovary, which is the basal bulge part, it is the basal bulged part where ovum or embryo sac is formed. Done? Did you understand till here? If you understand these two, male and female, double fertilization will be very easy for you. Done? Yes. So here, what we have to study, sir, stigma later we will discuss. Okay. First we need to study ovary. When we open the anther, inside the anther we were able to see microsporangia, Inside the microsporangia, sporogenous tissue, then microspore mother cell, then microspores, then finally pollen grains were there. Inside the pollen grain, two cells, very important, vegetative, generative, and generative produce two male gametes. Done? That was male. No? Now, if you see inside this ovary, if you take the structure of this ovary, this part, basal bulge part, no? Inside the ovary, we will be able to see first the innermost part. Okay? Inside the ovary, the first part is called as oviduct. Okay? Oviduct. Then it can also be called as cavity, ovarian cavity. Okay? Inside this ovarian cavity, we will be able to see the next layer, which is called placenta. Okay? Now, inside the placenta, we are able to see chambers like this. The chambers are called, yes, they are called as locules. They are called as locules. And inside these locules, we are able to see a small structures like this. It can be one, two, it can be many also. Okay? So that are called as ovules. They are called as ovules. Ovary, inside the ovary, ovarian cavity, inside the ovarian cavity, placenta, inside the placenta, locule, inside the locule, ovule. Okay? Now, another name for this ovule is called megasporangia. In the males, we have microsporangia. Okay? In biology, remember, whenever the word mega is used, that is used for females. Micro, micro kabhi bhi male, mega kabhi bhi female. Done? Yes. So, megasporangia. Done? Is that clear? Any doubts you have till here? Everything is clear. Shall we proceed now? Shall I raise? Huh? Then do it first. Done? Yes. So here, when we study the structure of the ovule, so here inverted structure anatrophus ovule. So here, if you see the structure of anatrophus ovule, outer integument, inner integument, nucellus, nucellus, then embryo sac. So here the point of attachment is funicle, which is connected to placenta, and the space is hilum. Yes. So here what we need to, मेरे diagrams होते अच्छे नहीं रहते हंसोना को, ठीक है? हाँ. So and that too in the marker. Uh, when we talk about the anatrophus ovule, what is this anatrophus ovule now? Ovule we got to know. Nahin, ovule we studied. Inside the locule, the ovule is attached. So here, if the ovule is attached in the opposite side, inverted, ulta, okay, that time it is called as anatrophus ovule. Inverted, we will tell. Two major parts. The bottom part is called chalazal end. Okay, and the point which we are able to see micropyle with the micropylar end. ठीक है, इसे चलाजा बोलते micropyle बोलते, where in the uppermost end micropyle is present, therefore micropylar end. Here it is in the bottom basal part, chalazal end. Is that clear now? So here what we need to study, how we studied the male gametophyte, right? Then we entered into the male gametes formation. Similarly here female gametophyte, then we have to go with the female gamete. Yes or no? Yes. So outer integument, inner integument, nucellus, then finally we have the innermost structure that is called as embryo sac, right? Two things we need to learn here. One is nucellus, one is embryo sac. Yes. Nucellus, what is the function of nucellus here? Yes. Outer integument is present, inner integument. Two layers are present. They are the protective layer. Okay. Inside that, one more layer is present. What is that layer doing? Yes. What is the function of nucellus? What it is doing? Why it is required? Nay, two layers were there. They are protecting the ovule. Then why one more layer is present? That is also protecting. Yes. If it is not protecting, what it will do? If it is not protecting, nay, it has to give nourishment. This is the nutritive tissue layer which will provide nourishment to the developing embryo sac which is present inside it. Okay. Now coming to the innermost part, that is embryo sac. What exactly is this embryo sac? Yes. What exactly is this embryo sac now? स्पेस बेस बेस इज चलाजल एंड. ये जो बेजल पार्ट है उसे हमें चलाजा बोलते हैं. ठीक है, चलाजा चलाजल एंड. Inside, innermost part is your embryo sac. What exactly is the embryo sac? Ovary, inside the ovary, ovarian cavity, inside the ovarian cavity, placenta, inside the placenta, we have lobes, locules. Okay, inside the locules, ovules. Inside the ovules, outer integument, inner integument, nucellus. Inside it, finally, end part. Okay, सबके अंदर we have embryo sac. This is called female gametophyte. Pollen grain is embryo sac is female gametophyte. Did you understand? Yes. Any doubt still here? Now this embryo sac, no, this embryo sac will undergo. It will undergo again meiotic division. Okay? How it will undergo meiotic division, sir? If you take the embryo sac, first this embryo sac will try to divide into two cells. It will try to divide into, so outermost, that is the nourishment layer. This is nucellus. Yes, and this again it will divide and form four cells. Okay, four cells. Yes, four cells will be produced. Now here what happens when the embryo sac is formed inside? No, how we have microspores, mother cells? How many were there? Many microspore mother cell. Now each microspore got converted into microspores. No, mother cell got converted to microspore. How many? Four. So one microspore mother cell gave rise to four pollen grains. No? But here what happens? This when the formation of this takes place, no, there we call it as microspore tetrad. No, here this are called as megaspore tetrad. Again it is tetrad. अगर ऐसा रहा तो हमें इसे टेट्राड बोले. सो नहीं, इसे क्या बोलना? लीनियर टेट्राड. It is linear tetrad. Now what happens after the four formation of cells, megaspore cell take place? Right? Three will degenerate. In this, three will degenerate. Okay? Only one will be remaining. One will become functional. There, each and every microspore mother cell was giving rise to the four microspores, and each microspore was giving rise to one pollen grain. Okay? But here what happened? When the megaspore mother cell gets converted to four megaspores, three will degenerate. Okay? One will be functional. एक ही काम कर रहा है, सो तीन degenerate हो जाते हैं. ठीक है? अब एक जो functional काम करता है, it will enter into the process called what? Which process? Don't know. Yes? Megasporogenesis? वो फॉर्मेशन ऑफ मेगास्पोर्स. अभी जो चार बने नहीं, इसे मेगास्पोरोजेनेसिस बोलते. सो एक मेगास्पोर मदर सेल अगर चार मेगास्पोर्स बना रहा है, that is only megasporogenesis. But what happened after megasporogenesis? Three megaspores, all the four, when they give rise to next four, it is correct. ठीक है? Then complete process हो रहा बोल को. But here what happened? Three functionless, they degenerated. एक ही चीज बाकी है, सो उन्हें फंक्शन कर रहा है. ठीक है? Now that one functional megaspore will get converted into embryo sac. उन्हें matured होको उन्हें embryo sac में convert होता है. ठीक है? Now what is that process called? Monosporic development. ठीक है? मियोसिस होता सब में नहीं, एक का चार हो रहा है तो मियोसिस. But the process, why it is called mono? Mono means single. Nay, mono means single. So only one is getting converted. So what will happen? In the monosporic development, one megaspore gets converted to embryo sac. Okay? Now in the embryo sac, if you take the structure of the embryo sac, if you take the structure of the embryo sac, first two cells will be present. नहीं, जैसे डिवाइड होती टू सेल फॉर्मेशन, एक सेल टू होता है, ठीक है. टू सेल्स विल बिकम, yes, two will become four, then four will become how many? Totally eight cell formation will take place. Yes. Now once the embryo sac have eight cell formation, that cell have to be arranged. Shall I erase? Mega, wait, what happened? Mega sporogenesis. This will lead to formation of monosporic development, and monosporic development will lead to the formation of embryo sac, and this embryo sac is the female gametophyte. Done? Yes. Yes. Now, so once the embryo sac is matured and all the cells, 8 cells stage that is seen in the embryo sac, that arrangement of cells will take place. So that 8 cells which are present, so 2 cells will be present. This end will be called as micropylar end, and this end is called as chalazal end. If you recall anatrophous ovule, in the anatrophous ovule chalazal end was down. Now it is, why? That was inverted, उल्टा. अब सीधा करे तो माइक्रोपेलर एंड नीचे आ रहा है, चलाजल एंड ऊपर जाता है, ठीक है. So two cells will be present, ठीक है. These two cells which are present are arranged in the micropellar end. Three cells will go and occupy the uppermost position. Then one cell will occupy here. How many? 1, 2, 3, 2 in the center. 8 cell stage is formed. But here, once the embryo sac is matured, we will not call it as 8 cell stage. ठीक है? 8 cell stage नहीं बोलते, इसे कितना बोलते? 7 cell stage बोलते. 7 cell 8 nucleate stage. ठीक है? Matured embryo sac will have 7 cell 8. हाँ, कैसा सर? देखो 1, 2, 3, 4, 5, 6, ये दो मिला को एक, ठीक है. 7, दो न्यूक्लियस अंदर हैं बट एक आउटर मोस्ट कवरिंग है, ठीक है. दो मिल को एक बन जाता, उसे हमें क्या बोलते हैं? ठीक है. सो हियर 7 cell 8 nucleate stage. Now the cells which has occupied the micropylar end, 2 cells which are present here, नहीं, इनको हमें synergid cells बोलते हैं. It is called synergid cell. One large cell, egg cell. Here is a central cell, ठीक है. So in that central cell, what is a primary endosperm cell with nucleus. 3 cells which are at the chalazal end, they are called as antipodal cells. Done. So egg cell and synergid cell together, it is called as egg apparatus. And here in the synergid cell, a small finger-like projections are present. They are called as filiform apparatus. It's a 3 marks diagram in your theory. Draw a labeled diagram of matured embryo sac. The matured embryo sac will have two ends. One is called chalazal end, one is called as micropylar end. In the micropylar end, three cells are present. Two cells are called as synergid cell, and one cell overlapping on it is called as egg cell. Okay? Egg cell and synergid cell together forms egg apparatus. Then in the center we have central cell with the primary endosperm cell and primary endosperm nucleus. Then on the chalazal end we have three cells which are occupying the anterior portion of the chalazal end, that is called as antipodal cell. This complete structure is called as matured embryo sac. Done? Did you understand? Yes. Draw it. If you know this diagram, okay, it will be very easy for you to explain the next process, that is called as double fertilization process. Angiosperms are the special plants. Okay? Other plants, when we see the bryophytes, pteridophytes, gymnosperm, there we are able to see only fertilization process taking place. But angiosperms are the special plants, okay? Advanced type of plants, highly complex plants with the double fertilization process C. Is that clear? If you take the angiosperms, almost 80% of the plants are angiosperms, okay? And more than 2,80,000 species of plants which we know, which we have studied, we have discovered, that comes under angiosperms, flowering plants. Okay? Yes. Any doubts here? Huh? Yes. Now, you are able to see female gamete here. Female gamete है इसमें. पोलन ग्रेन के अंदर मेल गैमेट्स मिले हमको. नहीं, कितने मेल गैमेट्स मिले हैं? 2 मेल गैमेट्स मिले नहीं. अब एम्ब्रियो सैक का फीमेल गैमेटोफाइट. अब एम्ब्रियो सैक के अंदर कितने गैमेटोफाइट्स मिलते हैं? फीमेल गैमेट्स मिलते हैं. Only that is X. Done? Yes. Now, if you see the male gamete, the male gametes are ready. Right? The male gametes, the pollen grain will come and sit on the stigma. Right? As I told you, the pollen grain, stigma is the landing platform for pollen grains. The pollen grain will come and sit on the stigma. Pollen grain will not enter. It will not enter. Right? When we studied the structure of the pollen grain, I told you when the pollen germinates, what will come out? पोलन अगर जर्मिनेट हुआ तो ट्यूब निकलता बाहर. नहीं तो अब ये पोलन अगर जर्मिनेट हो रहा है, इसके पास ट्यूब निकलता है, इसे हमें पोलन ट्यूब कहते हैं. नहीं, सो व्हाट हैपेंस, पोलन विल बी सिटिंग हियर ओनली. ठीक है, हियर when the pollen comes and sits here, that is because of a process called क्या अभी मालूम नहीं, शुरू से शुरुआत करना नहीं, एक साइकिल. पोलन ग्रेन अपने आप यहाँ को बढ़ जाता है, यहाँ को बैठने के वास्ते एक प्रोसेस है. नहीं, whenever the pollen grain is traveling, it comes in contact with the stigma. ठीक है, so from the anther, the transfer of pollen grains from the anther to the stigma of the flower is called, it's called pollination. नहीं, एंथर से पोलन ग्रेन निकल रहा है, उन्हें कहां जाना, स्टिग्मा के पास ही ना जाना, नहीं, मेल से फीमेल के पास जा रहा है, नहीं. What is that process called? It is called pollination. So with the support of pollination process, by the support of pollination process, the pollen grain will reach the stigma, and stigma is the landing product. अब कैसा आता सर? Who is carrying this pollination process? Any idea? Here? Pollinating agents. What are the pollinating agents? Yes? Water, air. Very good. Yes, water, air. Birds, insects. Anything else? That's all. Snails, beetles, honeybees, rodents, reptiles. Yes, there are so many. All the animals. ठीक है, zoophily का तो सब आ जाती नहीं. Yes. So with the support of pollinating agents, it has reached the stigma. Now, once it sits on the stigma, will the stigma directly give the permission to enter? Yes, there is again one mechanism involved. What is that mechanism called? जैसे यहाँ को बैठ रहा है, नहीं, स्टिग्मा जर्मिनेशन क्या, जर्मिनेशन किसे बोलते सो, ग्रोथ को बोलते सो. मीनिंग ऑफ वर्ड जर्मिनेशन इज ग्रोथ. अगर पोलन ग्रेन ग्रो हो रहा है, ट्यूब बाहर आ रहा है, उसे जर्मिनेशन बोलते सो. नहीं, स्टिग्मा is interacting. Stigma is interacting with the pollen. नहीं, when the pollen interacts with the stigma, कहाँ है सो, स्टिग्मा कहाँ है सो, स्टिग्मा कहाँ है सो, gynoecium composed of, gynoecium composed of stigma, directly stigma. क्या लिखाया था सो, मैं pistil or carpel, नहीं, gynoecium composed of pistil or carpel. Now pistil consists of three parts, stigma, style and ovary. अब pollen grain coming and interacting with pistil part, नहीं, pollen, pistil, interaction between them is called pollen pistil interaction. नहीं, one of the one important question, 1 marks का भी पूछ सकता है, 5 marks तक भी पूछ सकता है ये क्वेश्चन में. Explain the advantages of pollen pistil interaction or disadvantages of pollen pistil interaction, or what is pollen pistil interaction, explain. ठीक है, very important. So here what is happening, यहाँ क्या होता बोले तो, पहले pollination हो को pollen grain, it will come and sit on the stigma because it is a landing platform. Once it sits, permission is not given. Stigma क्या करता है, सबको permission नहीं देता. ठीक है, first it will interact. The stigma will interact with the pollen. So what interaction it will do, sir? Yes, it will check whether the chemical composition, whether the genetic composition needed is present or not. It is same or not. It is different but needed or not. So by studying the genetic constitution and chemical composition, ये दो चीज़ा, एक जो जेनेटिक मटेरियल है वो सेम होना, दूसरा केमिकल कॉम्पोजिशन सेम होना. नहीं, स्टिग्मा एक बार क्या बोलता है, नहीं, आई वांट डिफरेंट वन. सेम रहा तो अगेन इट विल गो विद द सेल्फ पॉलिनेशन, सेम क्वालिटी कंटिन्यू होता. नहीं, वी वांट न्यू. अब उसके वास्ते स्टिग्मा इज हैविंग द कैपेसिटी टू एक्सेप्ट द पोलन और टू रिजेक्ट द पोलन. ठीक है, अगर से स्टिग्मा पोलन को एक्सेप्ट कर दिया, नहीं, दैट पोलन इज कॉल्ड एज कॉम्पैक्टिबल पोलन. उसे क्या बोलती है, हमें कॉम्पैक्टिबल पोलन बोलते हैं. ठीक है, अगर एक्सेप्ट नहीं करया वैसे पोलन को हमें इनकॉम्पैक्टिबल पोलन बोलते हैं. डिड यू अंडरस्टैंड? यस. तो दो प्रोसेस खत्म होने के बाद एक पॉलिनेशन प्रोसेस खत्म होना, दूसरा पोलन पिस्टल इंटरेक्शन खत्म होना. आफ्टर दैट द पोलन grain will be sitting here. Ne? The tube will enter. Ne? The tube will enter. कहाँ आता है ने? First it will enter into ovary. ने? Stigma. Stigma के बाद style को जाता है. Style के बाद ovary को जाता है. Inside the ovary it will cross the ovarian cavity. Inside the ovarian cavity it will cross placenta. Nay, inside the placenta it will cross locule. Inside the locule it will enter, it will reach ovule. Inside the ovule, outer integument, inner integument, nucellus. अब nearby to the embryo sac. ठीक है. अब embryo sac यहाँ है बोले तो इन्हें दो मेल गैमेट्स यहाँ रिलीज कर डालता. ठीक है. जो पोलन ग्रेन के पास दो मेल गैमेट्स ही कहाँ रिलीज होती? Nearby the embryo sac. Is that clear? Did you understand? Any doubts still here? Yes. अभी हमें जितना भी पढ़े नहीं पूरा प्रोसेस एंजियोस्पर्म्स के अंदर this till here it is called as pre-fertilization event. क्या? हाँ, मेल गैमेट्स बना लिए, फीमेल गैमेट रेडी है ने, तो टुगेदर क्या ये प्री फर्टिलाइजेशन इवेंट. Done? Did you understand? Yes. Any doubts you have till here? Shall we proceed? You have drawn? Yes, draw it first. We will go with the very important process, that is double fertilization. It will be a 5-mark question for you. In your theory exams, 5-mark question. Explain double fertilization process. Bolko. Yes. This is the common topic both in first PUC as well as second PUC. We are studying plants. आप बनी पड़ी तो पढ़ाने वाले भी सही नहीं, पढ़ने वाले भी सही नहीं. फर्स्ट PUC में टेस्ट टाइप से क्वेश्चन टेस्ट है फर्स्ट PUC की है. किंगडम प्लांट है अपन को वो तुम्हें थियरी पता पड़ेगी. थियरी फर्टिलाइजेशन डायरेक्शन पड़ेगी तो थियरी ये जो मैं डिटेल में कह रहा हूं दिस इज बी कॉल्ड एज सीईटी. ये जो कंपनी है जो हमें पढ़ी नहीं पूछना है क्या करें फॉर्मेशन है जो बड़ा हो क्या है वो 20 रुपए सेकंड दैट इज एडवांस ये जो बेसिक पढ़ाई होती और एडवांस है. नेक्स्ट में करा मेरा नेगेटिविटी के फेसबुक पे ये एरिया सेंटर डेटा पे डबल फर्टिलाइजेशन का हम कवर एक डिफरेंट फैक्ट तो नहीं स्टार्ट करने में कोई बोला तो लेटर है ये देखिए लेटर स्टार्ट करने में कोई बोला नहीं दैट first we will go with the endosperm, then we will go with the development cellular particles. Yeah, why? Because it will be damage से हमारा है. अब हमें प्रोबेटिक प्लान मोड ये हेमोप्रोबेटिक प्लान मोड ये done. Shall we proceed? अजान के बाद अजान हुई नहीं अभी. अब जो नेक्स्ट पढ़ती जो है नेक्स्ट जो टॉपिक पढ़ते हैं दैट इज इंपॉर्टेंट फॉर द एरिया सेकंड डबल फर्टिलाइजेशन और एरिया साइड हमारे पास है. ठीक है, ऐसे भी पोलन ग्रेन से मिल गैमेट्स आपको क्या रिलीज हो. So next it will continue with the process that is called as double fertilization. Two times fertilization होता है वो double fertilization होता है. Explain double fertilization process in angiosperms. One of the 5 question. Expected question. Continue the vaga process. That will be good. For other topics, I give points. Because their points are different. Yeah, main and the stuff until you don't know, gamete में difference है. तो first thing बहुत मालूम नहीं रहता. इसे gamete बोलते हैं, इसे gamete बोलते हैं. वो भी इसे बोलते हैं. अब यहाँ में we are studying as egg. नहीं, in this flowers we are studying as egg. Right? अब if you go to the bryophytes, what नाम है? Anthrozoites. आर्चिगोनिया. ठीक है, वो भी हम फॉर्म करता है. Shall we proceed? So now we know the pre-fertilization event. Yes, shall we proceed with the second event? Post, directly post. Yes, now it is only fertilization event. Okay. Yes. So after the colon pistil interaction, after the colon, we'll finish this topic. Okay. After the pollen tube will enter into the stigma. It will cross the style part, then finally reaches the ovary, and inside the ovary, nearby to the embryo sac, it will release two male gametes. Yes. Now, the male gamete doesn't know where to fuse. Yes. So, with the guidance of the filiform apparatus, with the guidance of filiform apparatus, one male gamete will go and fuse with the egg cell. One male gamete will go and fuse with egg cell. So one male gamete, N cell. N cell. What will be the result? 2N cell. That will lead to the formation of zygote. That is diploid zygote. Yes. When the male gamete is fusing with the female gamete, the process is called syngamy. Or another word for syngamy is fertilization. डन होगा नहीं अब और एक मेल गैमेट बाकी है ना नहीं one more male gamete is present. So this one more male gamete again with the guidance of the filiform apparatus it will fuse with the polar nuclei. It will fuse with polar nuclei. Here how many nuclei are there? Two nuclei are there. So N cell, it will lead to the formation of Yes, 3n. That is triploid. Triploid endosperm. It will lead to the formation of ट्रिप्लॉइड एंडोस्पर्म. यहाँ zygote फॉर्म होता है, वहां एंडोस्पर्म फॉर्म होता है. ठीक, इन्हें 2n उन्हें 3n. कितने बार फर्टिलाइजेशन हुआ? 2 बार. नहीं, दिस 2n plus 1 3n formation, the process is called ट्रिपल फ्यूजन. इसे क्या बोलते हैं? Triple fusion. One time syngamy will take place, one time triple fusion will take place. So 2 times fertilization is happening. Therefore, it is called double fertilization. Yes. So this process of double fertilization was first studied, observed by a person named Navaschin. ठीक है, in the lilium species. It was studied in lilium species by a person named Navaschin. He was the person to discover this process called double fertilization. One time syngamy, one time triple fusion. Ultimately, the result what we have got is formation of zygote, formation of endosperm. Is that clear? Yes, this whole process is called as double fertilization. Done? Yes. Now, already you have studied in theory. Nahin. My question is for the next class. Okay? You should tell me the male gametes were left only in the chelazal, in the micropellar end. Assume if the male gametes are released in the chelazal end or it is released near the central cell, then what happens? Filiform apparatus is present here. Nay. Now, if the gametes were released on the antipodal cells, then also fertilization will take place. Yes? If it is taking place, how? If it is not taking place, why? This question will be your assignment for next class.